Thank you. Well, good, then they never heard me say anything about my cell phone carrier. That's even better. Thank you. So, okay. It's funny, I never got a chat message about that, but that's okay. So a hypertonic solution has a higher salt concentration simply because there's less water. A 10% salt solution is hypertonic to the cells because the cells have 99.1% water. 10% salt would only be 90% water. So what will happen is the cells will give up water if there's too much salt outside. And so the cells will shrink. Now, cells don't like to shrink either. Now, that can also produce some fatal consequences when we become dehydrated. Because when we get dehydrated, the first thing that happens is the water level in our plasma drops, which causes the interstitial fluid to drop, which causes the cells to give up water to go out of the cell to try and reach equilibrium, and the cells are going to shrink. And we can see that we call it crenation. We do it with red cells all the time. You've also seen it anytime you fail to water a plant, because apparently plants need water every so often, at least once a month. So you can tell I don't have a green thumb. So, but in our bodies, water movement is driven by the amount of salt. And when we become dehydrated, our salt levels rise outside the cells, not because we're gaining more salt, but because we're losing water. The amount of salt in our bodies is pretty constant, but it stays in solution at 0.9%. If we lose water, then the concentration rises, only because we've lost water. So we'll go into that in a lot greater detail in AP2 and fluid and electrolytes. I'll tell you now, there is no faster way to lose your patients. I mean, as your person you're caring for, than getting their fluid levels and their electrolyte, their salt levels messed up. Because once you enter that problem of trying to keep the salts up and the water higher, it can be a losing battle in here, and it can happen very quickly. Remember that we die without water after 3 days. We can't survive without water for more than 3 days. Okay, so the key terms here: hypertonic, isotonic, and hypotonic. So in hypotonic, water goes in the cell, and hypertonic, water goes out of the cell. So if you have to think of a plant that's not watered, it shrivels up because it's losing water. It's giving up water to the outside. And this summarizes the three terms: isotonic, hypertonic, and hypotonic. If we add too much water to a cell, it will rupture, it will lyse. And we hear the term lysis like in glycolysis, for example, when we're busy breaking down glucose, or breaking down glycogen, glycogenolysis, the rupturing of glycogen. Well, the cells can themselves just lyse, they can rupture. And red cells are very sensitive to that. So we never, ever administer distilled water to our patients. Now we can give a hypertonic solution to a patient if they are dealing with like congestive heart failure, where they have systemic edema, where they're retaining fluid in their tissues, and we want to get rid of that. We can also administer, there are various drugs that are diuretics, will get rid of that extra water too. So we don't want to retain water. We want to keep the water where it belongs. Okay. Now, we have active transport in our cells. We talk about all these passive processes, diffusion and osmosis, and facilitated diffusion and stuff like that. And we know our cells love to have that kind of movement. But sometimes we have to have what's known as active transport. Active transport requires energy. It requires us to pump against the concentration gradient. We know in diffusion, things move from the high concentration to the low concentration. It doesn't matter what it is. things will, passively move freely from levels of high concentration to low concentration. You saw the dye pellet. If I opened up a bottle of some sort of chemical up here, that's there's a chemical called isoamyl acetate, which is artificial banana oil. It smells like bananas. It's pretty good. And it's a little overpowering, but it's a pretty good representation of the odor of bananas. If I were to put a drop of that in a piece of paper up here, simple diffusion would say it would eventually disperse throughout the room and you would smell it. It's not an unpleasant odor. Butyric acid, on the other hand, smells like rancid butter. I could do that too, and it would go from here to here because the high concentration is here, the low concentration is back in the corner. And that's great. Simple diffusion goes from the high concentration to the low concentration. But in active transport, we have to go the opposite direction. It's like moving something uphill. It takes more energy. You just can't get water to flow naturally uphill. It doesn't do it. So with active transport, we have to pump things against the concentration gradient. Vesicular transport is even more complicated. Vesicular transport requires the use of vesicles, bubbles, if you will, that we can go into a cell with a vesicle or out of the cell with a vesicle. So let me show you what I'm talking about here. Okay. So active transport, we always go against the concentration gradient. We're moving uphill. It's like if you have if you got water in your basement and you pump it out, where's that water go? It goes into the ground. Where's it going to go from the ground? It's going to leak back in your basement, but you're pumping it out. There's more water in the ground than there is in your basement. So you're pumping, water's not going to flow out of your basement into the ground naturally. You got to pump it out. But naturally it'll flow. If you got too much water in the ground, it's going to flow right back into your basement. So anyway, with active transport, there's a couple things we consider here. We have two terms, one called the antiport and the other called the symport. Antiports have substances going in opposite directions at the same time. With an antiport, you're moving two substances against their respective concentration gradients. In our bodies, we like to keep sodium, high levels of sodium. We like to keep high levels of sodium outside our cells. We need sodium. We find sodium is present on the outside of all of our muscle cells, all of our nerve cells. Sodium, and on the inside of our cells, we have potassium. All of our nerve cells have potassium on the inside. All of our muscle cells have potassium on the inside. And the reason that we have a charge, an electrical charge on our muscle cells and on our nerve cells is because of the balance of the sodiums and potassiums. Well, there's so much sodium outside the cell that it will leak in. That's what it does. There's a tremendous amount of sodium outside our cells. It leaks into the cell. And there's a great amount of potassium inside the cell, and it will leak out because it's just, it's higher inside, leaks to the outside. Sodium's high outside, it leaks to the inside. We don't want that. If we allow a cell to continue to bring in sodium passively and lose potassium passively, that cell would reach equilibrium and it couldn't do its job. The ability for a muscle cell to contract depends on an electrical charge based on the difference between the sodiums and potassiums. Our muscles are electrical. They have a charge of minus 90 millivolts based on the sodium levels outside and the potassium levels inside. Well, sodium is always leaking in and potassium is always leaking out. Pretty soon we're going to have a charge in that cell of 0, and it's not going to function. So we have a pump built into the membrane of our muscle cells and into our nerve cells that pump sodiums back out. and it's going against the concentration gradient because the high concentration sodium is on the outside. Well, we're pumping sodiums back out against that concentration. And there's always going to be high potassium on the inside. Well, it's leaking out. So we're pumping the potassium back in against its concentration. It's like, you're at Walmart and you got your buggy and you turn down the wrong aisle and here comes everybody coming towards you and you're trying to go the opposite. We've all been there. So all of a sudden you're in the wrong aisle because everybody's going that way and you just want to go that way to get that bag of crackers. No matter, it's right there and I can't get to it, because you're into Walmart and see all the tourists standing around. And it's a family event. Picking out a bag of crackers takes a discussion from 10 people. You ever been there? Yeah. Okay. Home delivery from Walmart. Yeah.
The time of pumping the potassium, the sodium, which one is higher?
Well, there's always, that's really a good question. Sodium is always higher concentration than the potassium. We'll get into this in chapter 6, we get into muscles, but sodium is a positive ion, so is potassium. But there's a lot less potassium inside the cell than there is sodium outside. So we actually consider the outside of the cell to be positive and the inside negative, because even though potassium is a positive ion, there's a whole lot less of it than the sodium. So we say the inside is negative with respect to the outside. I know it doesn't make sense, even though we have two ions, they're both positive. Sodium has a positive charge, potassium has a positive charge, but there's probably 1 million more sodiums outside the cell than there are, for every potassium inside the cell, there's 1 million sodiums. They're both positive, but there's a whole lot more of them out here. So sodium's leaking in, potassium's leaking out. There's very little potassium outside the cell. That's why potassium leaks out. So, and you end up going against their concentration gradient. The normal flow, think of water going downhill. Well, you're trying to pump water uphill. So, and we don't like that. We have to spend energy. A cell will spend 40% of its energy budget just pumping stuff back and forth. Now, there's a term here called the antiport. Antiports pump sodium and potassium together, going in the opposite directions. When sodium leaves the cell, potassium is brought in. A symport is a pump or a mechanism that brings in two things together. Now, we're going to see a little later on that sodium has a best friend, and it's glucose. And when sodium comes into the cell, it will often drag glucose with it. And one of the things we do to keep glucose coming into our cell is that we keep pumping the sodiums out. It leaks back in, and we pump it back out. It leaks back in, and we pump it back out. But when it comes in, it usually drags a glucose along with it. So we see that sodium plays a role in bringing the glucose into the cell. Now, maybe it's because of insulin, setting up that special carrier protein that'll bring in sodium and glucose together, and that's okay. Because if we bring in excess sodium, we're just going to pump it back out. The glucose, we're gonna break down and release energy from it right away. So, but anytime we have active transport, we got to use ATP. That's the rule. Active transport doesn't work without ATP. Primary active transport and secondary active transport. Primary active transport uses ATP to get it to work. If we are going to pump sodiums out of the cell and pump potassiums back in, we're going to use ATP. Secondary active transport takes advantage of this. Secondary active transport doesn't use ATP, but it does take advantage of the fact that you pumped sodium out of the cell and it's going to come back in and it's going to drag glucose with it. It's a free ride for glucose. The sodium concentrations stay high because you pumped it out. You pumped it out. Okay. So where do we see this? This active transport, we see it in cell membranes all the time. We use, in our stomach, we have something called proton pumps that are pumping hydrogen ions out into the material in our stomach. Now, proton pumps in our stomach, lining our stomach, are why we have acid reflux. Acid reflux is based on high levels of hydrochloric acid in our stomach moving up against into our esophagus and burning the layers of the esophagus. And most of our reflux medications are what we call a proton pump inhibitor. They shut down the proton pump. You know, Protonics and omeprazole and Nexium, they all shut down the production of hydrogen ions. Because remember, hydrogen ions are what, you know, the more hydrogen ions you have, the more acidic the solution is going to be. An acid is something with high levels of hydrogen ions. And so the hydrochloric acid that we're producing to break down proteins in our stomach, we are overproducing those hydrogen ions. And so we take this medication as, you know, to treat gastroesophageal reflux disease, what we call GERD. And so these proton pumps affect lots of us. You know, if you're taking any kind of medication, famotidine, as I said, you know, famotidine, omeprazole, any of those, you know, so they all work as a proton pump inhibitor. Now, we have calcium pumps that we use all the time too. We use calcium in large concentrations to trigger the contraction of skeletal muscle. This is one of the reasons why we need lots of calcium in our diet, not just for our bones. It takes, you know, we have lots of calcium in our bones. It takes a long time for us to lose the amount of calcium that keeps our bones rigid. But we use calcium in our skeletal muscles to cause our proteins to slide together. Calcium plays a very important role there. And when we get a nerve impulse saying contract your muscle, we flood that muscle with calcium, the contraction occurs, and then we have to pump the calcium from around the muscle back into tubules that surround the muscle. And we're going against the concentration gradient there. We're trying to save that calcium. The most well-known pump, though, is called the sodium-potassium pump. We know more about that pump than anything else. The sodium-potassium pump is an antiport pump. It pumps sodiums out and potassiums back in. It always pumps 3 sodiums out of the cell and brings 2 potassiums back. Remember those numbers. Those are important numbers to remember. You know, I will confess, I don't think in any clinical setting anybody's going to come up to you and ask you, well, how many sodiums are getting pumped out? Well, you know, I don't think that's ever been discussed, but I know we know, you know, in clinically anyway, but we know that the sodium potassium pump pumps 3 sodiums out and 2 potassiums back in. And it involves gates and channels. Now you can see here that it's a carrier protein. It has a gate on one end and a gate on the other end. It has a gate inside the cytoplasm of the cell that allows sodium to go into the pump. And as soon as ATP comes along, which is the little blue structure down at the bottom there, the gate at the bottom will close and the gate at the top will open up, allowing the sodiums to leave. The gate won't open unless the ATP is present. As soon as the sodiums leave, the potassiums come in. Then the gate at the top shuts and the gate at the bottom opens and sodium goes back into the cell where it belongs. And we use, just on that structure alone, a cell will spend 40% of its energy pumping and pumping and pumping. And you can, this is nice. This is a decent little animation here you know, it changes its shape thanks to the ATP. It opens up one side, closes the other side, opens up and closes back and forth, but it's always 3 sodiums out and 2 potassiums back in. As long as the cell is functional, that's what it's going to be doing. Now, the secondary active transport doesn't require ATP. On the right-hand side of the picture here, we see the secondary active transport. We have pumped our sodiums out. We pumped our sodiums out and our potassiums back in on the sodium potassium pump on the left. But on the right, we have some passive carrier proteins that will let sodium come through, and some of them will change their shape. This carrier protein will change, when a sodium lands on it, it will change its shape to allow it to also carry a glucose. And so when sodium leaks back into the cell, it drags glucose along with it. And so we pay the price of an extra sodium we have to pump back out to get the glucose. Now, the good news is there's a tremendous amount of energy in every one of those glucose molecules coming in there. You know, so the cell can afford it. The cell can afford to allow sodiums to leak in if it's going to bring glucose with it. Okay. So that's active transport involving the sodium potassium pump. The other type of transport is using vesicles. Vesicles are bubbles. They're sacs. They're made up of the same phospholipid bilayer as the cell membrane. A vesicle is like a white blood cell. That's what you're seeing at the top there in the top video, is seeing a white cell engulfing bacteria, and it forms a bubble, a vesicle around the bacteria and brings it inside the cell. And once it's inside the cell, we can destroy the bacteria. And so the top video is an example of vesicular transport, engulfing something, bringing it into the cell. Now, there's 3 types. Let me rephrase that. There is 2 ways to move stuff across the membrane. There is endocytosis and there's exocytosis. Endo means bringing it into the cell. Endocytosis has phagocytosis, which is eating material, like eating a bacteria in the video on top, eating a cell fragment, because cells wear out and they break apart or they get infected. And so we kill them. We kill them and eat them. You know, so it happens. That's how we take care of our bodies. That's, you know, a cell is infected, we destroy the infected cell. The cell is old and breaking apart. We eat the fragments. We eat bacteria. We destroy viruses. You know, phagocytosis is cellular eating, and we engulf the material and form it, trap it in a vesicle, a sac, bring it into the cell, and then we digest it inside. Now, pinocytosis is cell drinking. We engulf something that is in some sort of fluid medium. Instead of just eating a fragment of a cell, we're eating, you know, something in solution. The same principle, we form a vesicle around it and bring it in. We never violate the integrity of the cell membrane because the vesicle is made up of a phospholipid bilayer, just like the cell. And when it comes together with the cell membrane, it fuses. And so the contents can be dumped into the cell, but the vesicle becomes part of the membrane itself. And then there's receptor-mediated endocytosis, where we're going after particular things. Now, exocytosis is leaving the cell. Now, watch what happens with the vesicle in the bottom. It comes up and it fuses with the cell membrane. So the cell membrane stays intact. Here's the cell membrane here. Here comes the vesicle, both phospholipid bilayers. They fuse together the contents are exposed to the outside, but you never expose the interior of the cell to the outside. The vesicle becomes part of the cell membrane, and then the cell membrane readjusts. So, endocytosis, the three ways we bring things in. We can simply engulf a large particle. This is like an amoeba does this with its pseudopodia, comes up and grabs it. White blood cells do the same thing. The cell membrane can grab a large particle and bring it in. And then it's inside its vesicle. The same thing with something in fluid. We form a vesicle around there. You notice how when it pinches off on the inside, it'll pinch off there at the top and it'll never expose the outside. And then a receptor mediated endocytosis, we're looking for something special. We're looking for something specific. Maybe it's an enzyme. Maybe it is a protein. Maybe it is whatever that we need, that we're looking for. And so only substances that will fit in that receptor site will be taken in. Exocytosis, we use this all the time when a cell makes proteins. When we make proteins, we bring a vesicle containing proteins to the surface of the cell membrane and we release them. And that's what it looks like. There's 100,000 magnification of a vesicle discharging its contents into the interstitial fluid around the cell. I don't know what it is. Might be a protein, might be a waste product, who knows? But that's how it does it. It fuses, and then there's no, the integrity is maintained. The cell membrane never exposes the inside of the cell to the outside. And this finally gets us into the structure of the cell. Now, the cytoplasm is what's inside the cell membrane. Has the consistency of pancake syrup, maybe really thin, watery pancake syrup. It's often been called cell sap or cytosol. Maybe a thicker, maybe it's more like corn syrup. But we've called it the cytoplasm, the cytosol. Old biology textbooks will call it the protoplasm. You go back about 60 years and you'll find a textbook that says protoplasm. And then gradually that changed into cytoplasm, and now cytosol seems to be the new term. All sorts of things are found inside the cytoplasm though. We have all the organelles floating in there. The organelles are the structures that do things for the cell. We find proteins, we find amino acids, we find glycogen and glucose inside here. There's all sorts of substances inside the cytoplasm. The organelles are going to be doing all the work for the cell, releasing energy from glucose, producing digestive enzymes, producing substances to destroy free radicals, things like this. So these organelles are very busy. Now the organelles can either be surrounded by a phospholipid bilayer, just like the cell membrane, or they can be totally unbound because of the way their structure functions. They don't need to have that phospholipid layer because they never leave, they're never exposed to the outside. So we refer to the organelles as either a membranous or a non-membranous. And the first thing we look at is the mitochondria. Every biology book tells you that the mitochondria is the power plant of the cell. And that's not truly correct. The mitochondria simply releases the energy found in the glucose. So we're converting energy from chemical energy into ultimately into mechanical energy. So we're taking the stored energy in the glucose and putting it onto an ATP molecule for about 6 seconds. That's just my prediction. You know, you can call it the power plant of the cells. It probably says that in the final exam about the power plant of the cells for mitochondria. Like I said, every textbook set calls that. I just don't like that term. The power plant?
Yep.
Yeah, see, there you go.
That's the only two things I can remember.
Well, there you go. So everybody calls it that. I just don't think that's accurate, but that's just me, you know. You won't go wrong saying power plant of the cell, because what it does is it takes glucose and it breaks the glucose down and releases all the potential energy held in the glucose molecule. And it only takes place in great numbers in the mitochondria. Mitochondria has a phospholipid layer. It has an outer layer and an inner layer, and all the work gets done on the inner layer. So we'll talk a lot more about that in chapter 24. So while mitochondria have this double membrane, ribosomes don't. Ribosomes are where we make proteins. Ribosomes come and go. They're sort of a transitory structure that forms and then breaks apart and forms and breaks apart. And every time it forms, it makes a protein. And then as soon as it's done making that particular protein, it breaks apart and then forms again to make another one. And it's always doing that because we're always making proteins. Now, ribosomes are made up of ribosomal RNA, that nucleic acid RNA. RNA comes in 3 types: ribosomal, messenger, and transfer. And the ribosomals make up the ribosomes along with some protein. We find most of our ribosomes attached to a series of tubes that surround the nucleus called the endoplasmic reticulum. We say these are bound ribosomes. Free ribosomes, which aren't nearly as many of, are going to be around to make proteins in the cytoplasm itself. Most of our ribosomes we consider are going to be bound to the ER. The endoplasmic reticulum, the ER, it's a series of flat tubes that surround the nucleus. Now, while it looks like the nucleus looks like a bowl and you have a series of tubes on one side, consider that the nucleus is shaped like a ball and you have a greater ball of tubes surrounded all the way around. Now, the rough endoplasmic reticulum is called that because it is studded with ribosomes. So all of our protein manufacture, our protein synthesis, every protein that we make takes place in these little purple dots there, the ribosomes. And we're, you know, protein for hair color, eye color, skin color, antibodies, antigens, muscle protein, any protein we make, all the hormones that are protein based are made in the ribosomes in this series of tubes called the rough endoplasmic reticulum. Now the other type is called the smooth ER. The smooth endoplasmic reticulum doesn't make proteins. Now it wraps itself around the rough ER. Let me go back and show you. There is the smooth ER right there. And there's the rough ER there with the purple dots. Now, the smooth ER does a whole lot of other things for us. It produces all the steroid-based hormones that are produced from cholesterol, like the sex hormones, testosterone, estrogen, progesterone. It produces all the adrenal cortex hormones like aldosterone and cortisol, where we get cortisone from, for example. It produces the non-specific sex hormones that come from the adrenal cortex, also the androgens. So all of these steroid hormones are made in the smooth ER. The lipoproteins that carry cholesterol, the HDLs and the LDLs, are made in the smooth ER of cells. We transport fats through the smooth ER. One of the most important roles of the smooth ER is detoxifying chemicals. We find in our liver cells we have many, our liver has many, many cells with smooth ER, more smooth ER in the liver than anywhere else in our bodies, because the liver is our detoxifier. Everything we eat, everything we drink has to go through the liver for detoxification before it gets to our blood supply. And that means before it gets to the cells. So whether it is a chemical in the food, a preservative, a coloring agent, a flavoring agent, maybe it's a residue of a pesticide that's below levels that might cause harm. Maybe it's medication. Like if you look at over-the-counter drugs or even prescription drugs, the dose of the medication is based on the age of the patient. You have a normal, healthy adult like us, and take Tylenol, for example. Tylenol, the dose for all of us for Tylenol would probably be 1,000 milligrams, 2 of those 500 milligram tablets. We all take it if you take Tylenol, or if you take Advil or something like that, it's 200 milligram tablets. That's the adult dose. Now, what will happen is that you won't get that full strength dose in your body because your liver is going to break part of that down. And whatever comes out of the liver is what our bodies are going to use to treat the pain or the inflammation or the fever or the headache or the backache or whatever we use it for. But that's okay, because the dose is based on a certain amount of it being broken down by the liver, and whatever's left over gets through is what treats us. So we pick the adult dose based on, what's going to come through the liver, because the liver is going to try to clean it up no matter what it does. If you were to give a child's dose to an adult, it would have no effect, because the child's dose of Tylenol would never get through the liver. The liver would break it all down. And you wouldn't give an adult dose of Tylenol to a child. 1,000 milligrams is way too much for a child's liver to process. That's why we have doses based on age and body weight. Body weight has a whole lot more to do with it than age, because the body weight deals with the mass, the stature, how well can we process this stuff. Just like an 85-year-old may not get 1,000 milligrams of Tylenol either, because their liver is older. Over here, their liver is older and may not be as efficient. Can they handle 1,000 milligrams? Maybe they can only take 500 milligrams. And maybe their liver can't deal with 1,000, and 1,000 is too much. So our liver is busy doing all this detoxification for us in the smooth ER. Alcohol is a good example of what our liver does for us. Remember, everything we eat and drink goes through the liver first. For every ounce of alcohol a person ingests, the liver is going to break it down into carbon dioxide and water. That's the end result of all metabolism in our bodies, is always carbon dioxide and water and maybe a release of energy. So a person ingests an ounce of alcohol, that is 1 beer bottle or 1 can of beer, that is 1 mixed drink or 1 glass of wine. So all 3 of those, the key term here is 1 ounce. And it will take the liver an hour to process that 1 ounce of alcohol. It breaks it down ultimately into carbon dioxide and water. It goes through a whole series of chemical reactions. Some of the byproducts that form are very dangerous, like formaldehyde, but as it gets formed, it's immediately broken down. No problem. Well, if your patient confesses to drinking more than an ounce of alcohol an hour, the liver can't keep up. Okay, so they had 2 beers in an hour. Okay, I've heard that people do things like that. I'll just let it go. The more alcohol you ingest, the harder it is for your liver to keep up. And one of the consequences of
People drink 2 more than 2 years now.
Yeah, I mean, I'm pretending that this is not realistic, but the more alcohol we drink per hour, the harder it is for our liver to keep up. And so if you drink a lot, if you ingest a lot of alcohol over the course of an evening, more than one ounce per hour, then your liver is still going to be breaking it down, but you're going to feel bad the next day based on how much you've consumed, because it takes time to break down to form these waste products and then break them down. So if a person drank a lot of alcohol over the course of an evening, the next day they're going to have what we call the hangover. And the hangover is simply the body trying to continue to process all these toxins that were formed as the alcohol is getting broken down. That and the fact that alcohol also inhibits your ability to regulate your fluid levels. In other words, the more alcohol you drink, the more you have to pee. And the more you pee, the more dehydrated you become. Because when someone is recovering from the night before, in addition to feeling bad, they're also very thirsty. And because they've been peeing all night long. Hopefully they've made it to someplace they can get rid of the urine without just letting it go. But that happens too. Now, realistically, I would imagine that 1 ounce of alcohol per hour sort of takes the fun out of drinking it, if you will, because if you have an ice cold bottle of beer and you let it sit for an hour, it's not ice cold anymore, and it's probably flat too. So anyway, something that when your patients say, "Oh, I feel terrible," well, why? Because I drank a lot of alcohol, a lot of beer last night. Okay, fine. I would imagine there's probably a lot of people in Knoxville on Sunday morning that had some issues, So now, so that's a big thing what our liver does. That's an extreme example because lots of people don't overindulge in alcohol every night. Some do. You have your chronic alcoholics that are going to be doing that. I guess it's sort of redundant. But when someone does tie it on, now the problem comes in particularly when you drink so much alcohol, the liver can't keep up, and it keeps making these toxins. It's trying to break them down, and your patient dies of alcohol poisoning. That happens every so often too. Since we live so close to a university, every year or so you hear about some freshman that dies because they never been exposed to drinking before, and they didn't know anything about moderation or what will happen, and they just kept drinking and drinking, and then they die. And it happens. It doesn't happen very often, but it does happen. Now, what else does the smoothie R do for us? Glycogen, the stored polysaccharide in our bodies, the polysaccharide that we use in lieu of starch. We break that down to glucose, and that takes place in the smoothie R. And all that calcium that we need to have muscle contractions, our muscle cells have smooth endoplasmic reticulum wrapped around each cell, packed full of calcium. And when we want to contract that muscle, we will release that calcium to flood those muscle cells, and the muscle cell will contract. So we store the calcium in the smoothie R for us. Okay, Golgi apparatus. The Golgi apparatus is another series of tubes or sacs that surround the rough ER. And what happens is when a ribosome makes a protein, 10 million of its closest friends are also making proteins. They're all different. because we have these millions and millions and millions of ribosomes around the nucleus, and the instructions have come out from the nucleus: make hair color, eye color, skin color, hemoglobin. Let's make a couple of dozen hormones. And so you have all these different proteins being made. And when they're leaving the endoplasmic reticulum, they just pack everything up into a vesicle. Cells like vesicles. They pack everything up into a vesicle and ship it off to the Golgi. The Golgi takes that vesicle, takes all the proteins out and sorts it out. So that you get all the hemoglobin in one vesicle, and all the hair color in another vesicle, and all the skin color in another vesicle, and all the digestive enzymes in another vesicle, and then they release those individually. So you don't just open up the cell membrane and release a bunch of stuff. You organize it. Sort of like FedEx. When FedEx first started, they started out of Memphis. Did you know that? Yeah. FedEx started in Memphis, and that was their central hub. And all the FedEx planes from all over the East Coast and the West Coast fly in there. And then there's this mile-long sorting facility. The planes would pull in, and they unload their, the nose of the plane would pull right into the front of this building, on the side of the building, and all the cargo would come out. And it would run through these huge conveyors and be sorted. So you'd have a plane going to Chicago and Denver and Seattle, or New Orleans or DC or something like that. And all the packages would get sorted right there. There they go. Now they've got all sorts of regional centers that do that for them. But that's how they started out, from right there. And that's what the Golgi does. The Golgi takes everything that comes in and sorts it into the individual vesicles. UPS does the same thing in Louisville, believe it or not. So, okay, so what it looks like this. The Golgi apparatus, a series of tubules. A vesicle comes in, a transport vesicle comes in from the rough ER containing a mixture of proteins. And it fuses with the Golgi. And then all the contents are sorted out. So you get all the hemoglobin in one and all the digestive enzymes in another. And then you have these individual vesicles coming off the opposite, the other end of the Golgi, releasing the same protein. And everything is sorted out. So if we want to have hemoglobin being made, we're going to get nothing but hemoglobin vesicles coming out. And we're going to get nothing but pigmentation proteins, and we're going to get nothing but growth hormone, and we're going to get nothing but melanin proteins coming out. We're not going to have just a collection of it. And this is a great picture here because it shows you the ER with the vesicles coming off the rough ER, and then we have actually 3 different pathways. When we get to the cell membrane, we're going to release a protein, like you see at the bottom. We're going to incorporate that protein into the cell membrane. Perhaps it's an enzyme. Perhaps it's a reactive site enzyme. It's going to stay there. And that's the second pathway. The third pathway is to keep it inside a organelle called a lysosome. Those are the digestive enzymes. So we can do 3 different things with these proteins. We can release them, we can fuse them into the membrane, or we can store them inside the cell for later use. And it's just fascinating how we just keep sorting all the time. Okay. One of those vesicles that can get formed is something called a peroxisome. A peroxisome destroys a free radical. A free radical is a molecule or a fragment of a molecule that occurs during metabolism. You're breaking down glucose to release energy, and you get a fragment. Perhaps you get a sodium atom that comes off, and instead of becoming like a sodium ion where it lost an electron, perhaps the sodium loses a proton, And now instead of having sodium with a positive charge, you have sodium with a negative charge. You're not supposed to have negatively charged protein and negatively charged sodiums in your body. Or chlorine, like sodium chloride, only chlorine has a positive charge. Instead of picking up an electron, it picks up a proton. That's not supposed to happen. These are what we call free radicals. They are oxidizing agents that cause the cell to break down other structures that we're not supposed to, and they're very dangerous because a free radical can run through the middle of a cell and damage other structures. So we want to get rid of it. We want to use an antioxidant, if you will. We hear about, some of the, it's good to eat antioxidants, like, dark chocolate, for example, is an antioxidant. Red wine is an antioxidant. The leafy greens are antioxidants. They break down these free radicals. In addition to that, because we don't want them in there, it may be that that free radicals are cancer-causing agents. Our cells break them down too using peroxisomes. There's two enzymes involved here. Oxidase, whatever the free radical is, oxidase will break it down to hydrogen peroxide. Same stuff that we go down to Walgreens and you buy a brown bottle, and you pour it on a cut and it bubbles out, and it doesn't hurt. When you're a kid, you remember when you had a cut and you poured alcohol on the cut and it was like a new experience in pain, but hydrogen peroxide just bubbled and it didn't hurt. And it did the same thing. Well, this hydrogen peroxide is a little stronger than that. This hydrogen peroxide is very dangerous. So we break the hydrogen peroxide down to water. We use a second enzyme called catalase. So we form hydrogen peroxide, then we break it down to water. And so that gets rid of these free radicals. There's a little bit they do with fatty acid production too. So, but we'll get more into in breaking down fatty acids in AP2 when we talk about diabetes. But the main thing they do is get rid of these free radicals that can form. They can form naturally simply through, they're a byproduct of metabolism that we shouldn't see there. Now, this other type of organelle are the lysosomes. The lysosomes are the digestive enzymes that I've been talking about. A lysosome is nothing but a sac of digestive enzymes, and it contains the enzyme because we don't want to release this digestive enzyme into the cell because it would destroy the cell. So we keep it inside its own little acid-proof vesicle. The only time we use this is when the cell eats a virus or eats a bacteria or eats a cell fragment, and it decides, okay, I'm going to fuse my lysosome with the vesicle, the cell fragments in, and it can dissolve it. That's what it does. Anything that doesn't work right, anything that's worn out, anything that's broken, we use lysosomes for. We use it to break down glycogen. We use it on our bones. When our bodies need more calcium, we will start dissolving bone using lysosomes. And it just dissolves away the organic matter, and the calcium gets released, and our blood calcium goes back up to where it belongs. If a cell is damaged beyond repair, the cell will undergo self-destruction, what we call autolysis. The cell will release its own digestive enzymes and dissolve its cell membrane and fracture, essentially. And then if that didn't take care of the cell, then all its friends and neighbors will come over and finish the job. Lysosomes play a really important role for us in protecting us. So we're always making these digestive enzymes. It's how white cells will engulf a pathogen and kill it. There's a whole type of white cell called natural killer cells. And what they do is they will surround and engulf a pathogen of some sort and then break it down. Now, there's something special about lysosomes that we pay attention to in the Southeast. There is a condition known as Tay-Sachs disease. Now, when we are born, our nervous system, our central nervous system continues to develop and continue to develop and grow. And as it does, we form protective layers around the central nervous system called glycolipids. You know, it's like a sugar-coated fat with like a glucose, you know, a carbohydrate and a fat together. Well, we don't want to have, you know, too many of these special glycolipids forming here, because if they grow uncontrollably, they start squeezing the brain matter and squeezing the spinal cord, so it causes brain damage. And so there are lysosomes that specifically break down these fatty glycolipids, you know, not enough to cause any damage, but just enough so that they don't accumulate. You know, if some is good, more is not better. We want to keep the glycolipids in check. They're doing their job, protecting the brain, protecting the spinal cord. We just don't want them to overgrow. So in Tay-Sachs, which is a homozygous recessive trait, meaning that you have to have two recessive genes to have the condition, meaning it's an inherited disorder. The mother and the father will contribute the recessive gene to their offspring, and if the child receives the recessive genes, they'll have this condition. Now, it depends on the severity, but many of these cases, they're all focused on Tay-Sachs disease does not have the lysosome present to break down the glycolipids that accumulate. So what'll happen is a child with Tay-Sachs disease, an inherited disorder, will start accumulating these fatty deposits in their brain without having them broken down, and it will start compressing the brain and compressing the spinal cord, and it usually kills them by age 5, and there's nothing you can do. Now, there are variations. There's variations on anything. In some individuals it starts later, some can even start as a young adult. Those usually don't have bad consequences, because by that point, once we're past about age 16 or so, our brain and spinal cord is fully formed. So we're pretty safe at that point. So now, why do I mention this? Well, it was primarily contained in some very distinct population groups. Most people that carry the Tay-Sachs gene came from descendants of Central European Jews. You know, and if you know anything about European history, you know that Jewish people lived in very closed societies, and they married within their society, keeping that recessive gene alive. You know, so it was always there. The reason Mediterranean Jews, you know, from the Middle East don't have it is because they married everybody. And so they diluted these recessive genes. So it's very rare. Be that as it may, most of your cases of Tay-Sachs show up in descendants of Eastern European Jews. Now, there is also a subset of people of French descent, but it's the French descent were settlers of Eastern Canada back in the 1700s in Nova Scotia, in that area. Well, so they were French, and when England took over Canada, this is not a history class, but you know, and there's a point to this. When England took over Canada, everybody, all the French settlers in Nova Scotia, the French Canadians, had to leave. So where did they go? They went straight south to Louisiana, because that was French territory in those days. They settled below New Orleans and Baton Rouge and places like that, and they're known as the Cajuns. They call themselves Cajuns. They carry that gene too. They carry Tay-Sachs, and their offspring to this day will still have Tay-Sachs. Now, why do we talk about that? Because Louisiana isn't that far from here. It's like 8 hours away. And when there's a hurricane, like, remember Katrina? You know, you heard about Katrina probably years ago. There was a mass, I was teaching here when Katrina came through. So it wasn't that long ago, but there was a migration of people from Louisiana because of the damage caused by Katrina, and many of them stayed here. And so it's entirely possible that some of them, it's logical to conclude that some of them have this gene. So you're probably more likely to encounter Tay-Sachs here than you'd ever expect. Just something to keep in the back of your mind. You know, it's just one of those little off the wall type of diseases, but it affects the lysosomes. Well, on that cheery note, let's move on. So I'm full of all sorts of cheery stuff this morning, aren't I? So the cytoskeleton. Now we know our bodies have a skeleton. We're looking at it right here. We've been talking about it in the lab for 3 weeks. We also have a skeleton at the cellular level made up of filaments. Because why do we have the shape of certain cells? Well, it's because of these filaments that give it its structure. We have cells that are long and skinny, nerve cells that extend for 3 or 4 feet in length. The sciatic nerve, I think I showed you that. Everybody, I showed that. I showed it to the Thursday lab because I was using the anatomage. I'll show it to the Tuesday and Wednesday classes this week too. Finally got that thing to work for us. Sciatic nerve is made up of thousands of individual cells that run the length of our leg. Why are they long and skinny? Why are red cells shaped like a donut? You know, why do amoeba or white cells act like an amoeba with this squishy membrane? It's because of their cytoskeleton. We have filaments, microfilaments, intermediate filaments, microtubules, all doing things. They act like the ligaments and muscles in our body, but only on the cellular level. So the 3 components of our cytoskeleton are the microtubules, the microfilaments, and the intermediate filaments. So for example, what is a microtubule? Well, spindle fibers. We've all seen pictures of spindle fibers during mitosis, pulling the cytoplasm, pulling the chromosomes apart. Those are nothing but tubules. It's thought that the way that the fiber moves is by taking those little fragments off of the end of one end of the tubule and adding to the other. So the tubule seems to move as it's really changing its shape. The second thing are the microfilaments. These are contractile. Actin. Actin is a contractile protein. It's a microfilament that enables it to slide over top of another microfilament so a muscle can contract. And then the third type are the intermediate filaments. We often see these as cilia and as flagella. So all 3 of these together give our cells their definite shape and its movement. I mean, some cells move, sperm cells move. They're the only cells in our bodies that actually have mobility to move around, to other with a purpose. White cells float. They can change their shape, but they float. But a sperm cell actually moves. Okay. Something else we encounter. We have what are called centrioles. Centrioles is where we form the spindle fibers from. They set in the cytoplasm just below the nucleus. And when we undergo mitosis and cell division, they will double and they will start and they'll move to opposite ends, and the spindle fibers will start there at these centrioles. And they are microtubules that will create these fibers for us. Okay. Now, something else we see besides the cilia and the flagella, I just mentioned those. Cilia don't push the cell, but they do move things. Cilia will move mucus up our trachea. You know, our airway, we suck in dust and dirt. It lands in the mucus lining our trachea, and underneath the mucus are those cilia on those pseudostratified ciliated columnar epithelial cells. The cilia push the mucus up and we swallow it. We swallow about a pint of this mucus a day. Fortunately, we aren't aware that we're doing it. And it's because it's all full of crud. We have cilia that line the oviduct in the female that pushes the egg along. Whether it's fertilized or not, it's still pushing the egg along. It takes the egg 7 days to go 2 inches because it's being pushed by these little cilia. Something else we do is we also increase our surface area. We use villi. Villi is Latin for fingers. Microvilli means tiny fingers. And villi are projections that stick up off the surface of the cell membrane. And microvilli then stick out at right angles from the villi. And what that does is it increases the surface area. Our small intestine is only 20 feet long. It's no bigger around than maybe your thumb. What if you were to spread out the small intestine because flat to encompass all of the projections? See, the small intestine is made up of simple columnar cells with villi and microvilli. The total amount of surface area in your small intestine would likely cover this classroom, the hallway, and the classroom next door. There's that much space for absorption in there. Because this 20-foot long little tube doesn't, just like that, doesn't give you a whole lot, but if you stretch it out, it's a huge amount of space for absorption. Okay, this is a great place to stop. We'll pick up with the nucleus on Wednesday. Lab this afternoon, if they thaw out. Wait, no, lab this week is going to be, I almost told you about the hearts. Lab this week is going to be joints and articulations, and we'll be using the anatomage. So if you're online or in classroom, we're doing this. It's too bad you're not in A&P2 because we are dissecting deep parts today. So, assuming that they so, I may have to mark it. So, but anyway, I will see you all on Tuesday, at least for lab, and I'll see you lecture on Wednesday. And thank you for your patience with